Welcome to our webinar, Camp Series webinar with Nissa Coit. She is now, oh gosh, you're a grad student. You've, I think you're done your thesis, right? Has it been accepted?
I haven't actually filed it yet, so
Okay,
it's done, but like not officially. So I don't have the diploma yet, but not far off.
good. Well, congratulations. And Nissa is going to be sharing that incredible work with us this evening, the effects of EO on honeybee overwintering. And we'll all be more in the know at the end of her webinar this evening on the effects of EO and what exactly is EO, right? Nissa has been with the Nino Bee Lab now for 3 years. Yeah, and has become and always has been since her beginning with the lab, an integral part of our team. Nissa, you might recognize from some of our honey and other products of the hive class. Nissa's quite crafty with all things honey, wax, and even wool, crewel work, and I don't know. You're always working on something, which is this is great. It's great to see what you can create, and we look forward this evening to hearing what you have been working on over the last 3 years, Nissa. Welcome
Yep.
this evening.
Thank you. All right, are we ready?
We are. Take us
Okay.
away.
So I actually just finished analyzing the last of the data last night. So this is fresh off the presses. And we'll get into it, what I found. So let's see, the effects of ethyl oleate. So basically the question that I had that got me started on this whole thing is the question of how do winter bees overwinter and what causes it. So if you aren't already familiar with the way that overwintering works in honeybees, they enter this what's called diutinous or long-lived state in the winter, where they can live for up to like 20 weeks, which, as all of you probably know, is pretty unusual compared to summer bees, which can live for only 6 weeks. And they are, during that time, they cluster together for warmth, and the queen stops laying eggs, and they are just the same bees making it through the winter. So there's not that continual population turnover that you see normally with summer bees. So we don't know exactly what triggers it, and we're not entirely sure like which bees go into this overwintering state. So like whether they're nurse bees or foraging bees, or what the demographics are of the overwintering population. So it's really quite a mystery. We don't even know exactly what all the environmental triggers are. So some of the proposed ones obviously are cold, colder temperatures, day length, so like photoperiod, pollen dearth, and other sociodemographic cues like pheromones, caste division, the number of bees present. And so it's probably a really complex system of things that cause it, but we're not even sure like what some of those factors are, because photoperiod by itself doesn't seem to be enough. Pollen dearth seems to be pretty good. So in order to answer a lot of questions about wintering bees, I needed to figure out what exactly was a winter bee and what caused it, because there's a lot of other questions about management that can't be answered until after we figure out more about it in this regard. So the physiological attributes of winter bees are that they're really similar to nurse bees in that they have large hypopharyngeal glands, which are the brood food producing glands in the heads of the nurse bees. Well, they're in all of the bees, but they're developed more in the nurse bees. They have low juvenile hormone, and they have high vitellogenin, which is associated with fat body storage and reproduction in other animals. They also have possibly larger fat bodies and increased immunity and different metabolisms. So we thought maybe that something about their immune system changes in the winter, which allows them to live longer, or that their metabolism is different. But none of these questions were answered, so I tried to sort of isolate that. The other thing about the winter bees is that while they have large hypopharyngeal glands resembling nurse bees, they're actually not active. They're not actively producing the proteinaceous secretions, the royal jelly that they are usually creating. So they're big, but they're not like active, they're not full of protein, they're like empty. So that's a big distinguishing factor between winter bees and nurse bees. And there's also evidence that in normal summer bee behavior or summer bee populations, young forager bees are able to revert back to a nurse bee state when they have removed the nurse bees. So there was one study that happened when they removed all the nurse bees and the young foragers were able to develop their hypopharyngeal glands again and start nursing the brood. So there's evidence that they're able to sort of revert this way. And I was wondering if that kind of reversion could also happen with the winter bee state. So if older foraging bees could then revert back to winter bees, or if only nurse bees were proceeding into the winter. So to continue, like I said, photoperiod doesn't affect nurse bee, doesn't affect overwintering, but it does, however, affect how plants are able to bloom and create pollen. So in the autumn period, the bloom might be reduced, so there might be less pollen available, and so bees might be foraging less. So that might be sort of an indirect cause of shorter days in the fall and winter in impacting the transition to overwintering. So we do know that pollen feeding triggers prolonged brood rearing. So if you continue feeding your bees later into the season, they're going to continue producing more and more brood. And this brood rearing actually delays the onset of diutinous bees. So bees that have been fed pollen for longer into the winter are also going to create winter bees later. And this transition, when it's not lined up correctly with the actual environmental cues, can kind of impact their ability to come back at the end of the winter, in fact, which bees enter the winter bee state. So kind of manipulating their pollen supplements can potentially lead to reduced winter survival, or at least in the ability to overwinter correctly. And also, like I said, the diutinous bees do not become present until after brood rearing stops. So you don't want to continue creating brood by feeding pollen if you want them to overwinter successfully. The other thing is that brood rearing reduces the amount of vitellogenin stored in the fat bodies, and it reduces the colony's overwinter success and extreme longevity. So it's sort of like as they're rearing more brood, they're not able to put any pollen that they've consumed into their fat bodies and like create winter stores. Like if you imagine a bear that's getting ready to hibernate, if they're like breastfeeding, for example, they're not going to be able to put those stores to their own body and able to overwinter. So this is sort of the mechanism that I was trying to focus on to determine how overwintering happens and what causes it. So I focused on one particular pheromone, ethyl oleate, which is what EO stands for. And in a normal summer bee population, it's responsible for delaying the onset of foraging. So if there's a bunch of foragers out bringing in pollen and nectar back to the hive, they don't want more nurses to age into that role if they have enough already, because they want to create this balance of proper age polyethism and dynamics of division of labor to match the incoming food reserves and incoming food availability. So basically what happens is this ethyl oleate is produced when forager bees consume fermented nectar and ethanol from that fermented nectar. And when they're getting enough nectar coming in, that they produce more of this ethyl oleate, and the ethyl oleate tells the nurses basically not to age into foragers. So it keeps them in this young nurse bee state for longer. And I thought that this particular pheromone would be a good candidate for creating this nurse bee state in winter bees for a variety of reasons. So to continue on the topic of ethyl oleate, the high juvenile hormone levels in forager bees transport ethyl oleate to the honey crop, and it's exuded through their exoskeleton on their abdomen, where other bees perceive it by antination in close range. So it's not volatile, which means it doesn't just like pervade through the colony. It's perceived through close contact, which is going to be important in my experimental design later on. Sorry, there's a kitty. So the basic like hypothesis that I had was that the high concentrations of ethyl oleate would be increasing with the seasonal changes as forager bees were staying in the hive longer due to either poor weather like rain, shorter days, or just not having anything to forage on. So I thought perhaps that ethyl oleate could keep the nurse bees in a nurse bee state longer and help them transition into the winter bee state. The other question that I had was whether that effect could also cause forager bees to revert back to the nurse bee winter bee state, like was seen when they had the necessity of doing so to feed brood when they removed all the nurse bees. So we're not sure if the reason that they reverted in that situation was because of ethyl oleate, but it seemed like a good candidate because of the common factors between all of these physiological conditions. So let's see what my experimental design was. There we go. Basically, I covered this a little bit. As the days shorten, the temperature drops, the weather worsens, pollen becomes less available, the forager bees are spending more time in the hive, and they have more exposure to ethyl oleate, both forager bees and nurse bees. And we wanted to know if it worked on forager bees and nurse bees in the same way, or if one of those age groups was sort of more behaviorally plastic in ability and able to respond to ethyl oleate better than the other one. So we're looking both at whether ethyl oleate contributes to the physiological effects of winter bee and which age groups it works best on. And also this kind of translates into whether true overwintering can happen in moderate climates like here in Northern California, because in Southern California and other tropical areas, bees don't really overwinter. And then as we know in Northern climates where it's really snowy and cold, they definitely do. But we're in this area of this sort of gray area where we don't know whether they're actually overwintering or not. And it was really hard to answer that question without knowing more about what is a bee, what is a winter bee, and whether they like what causes it. So that's sort of what we're trying to get at eventually. That's not really covered in this particular experiment, but it's sort of laying the groundwork for it. We also want to know is if you're not properly recreating the demographics of the overwintering bee population, if that's going to impact overwintering success. So if it's supposed to be mostly nurse bees, or if it's supposed to be mostly forager bees, or if it's supposed to be a mix, and you're feeding pollen later into the season, or you're giving them ethyl oleate, for example, if that became a part of the technology, or if you're putting them in cold storage, is that going to be affecting the way that they overwinter? But we're not really going to cover that here. So these were just sort of overarching research topics that could be answered eventually. So what we're basically looking at is what factors contribute to overwintering and how ethyl oleate affects those physiological markers. So this is a really complicated sort of map of all of the different things that potentially affect overwintering and the transition both into overwintering and out of it in the spring. And I'm not going to go into the details of it, but ethyl oleate sort of is just this one little piece here. So the experimental design that I created in order to answer some of these questions was basically I painted emerging day-old bees from a single colony, which I've shown here as the donor colony. And the bees actually emerged in waves. So each day that they emerged, I painted them a different color, which was a logistical necessity because I could only get 1,000 bees from one colony to emerge on a given day. So when I painted the bees, I inserted them into another colony, which was in a tent, and I put them in a tent so that the bees that I painted wouldn't drift to other neighboring colonies, because we have a fairly large apiary, and I didn't want all my hard work to just be lost. So I put them in a tent to sort of know where my bees were, which is the foster colony. And I ended up putting like 3,000 painted bees in there. So on each, the point of painting them was so that I would know how old they were. So on the first day I painted 1,000 bees green, and I knew that if I went back in 7 days and I found green bees, those bees would then be 7 days old. So that's exactly what I did. On the 7th day, I went in and I removed a subset of the bees that were in the cage, in the hive, and I put them into 4 different cages. So the different cages were ethyl oleate treated and control treated. And I actually fed them the ethyl oleate in different ways. I mix it into a candy fondant, and I also just put it on filter paper because I wasn't entirely sure how they were going to perceive that and whether they would actually eat the candy. So I wanted to make sure that they were getting it. And even though ethyl oleate is not a volatile chemical, I didn't want any of it to affect my control bees. So I put them in separate incubators at the lab. So that's what these boxes here represent, is them being in separate incubators. So the 7-day-old bees that were put into these cages represented nurse bees. And then on the 21st day, they represented what I called young foragers. And on the 28th day, I also got old foragers. And the reason I did that was because from the other study where they removed the nurse bees and had the foragers revert back to a nursing state, the bees that had not been foraging for very long were best able to revert. So they still had the ability to sort of swing both ways, and they had this behavioral plasticity. So I kind of wanted to look at both of those elements. So I did that a number of times to get a 5 times replicate. So I had 5 of each of these. And then I sampled them. So this is me. You can see how many different colors there were on one frame. They're in this bucket before I inserted them into the colony. This is the tent where the colony was. When I actually went to sample the bees out of the tent, actually, I wanted to get them actually foraging, because the foraging bees, or the division of labor, the difference in actually performing these different tasks has a greater impact on physiology than the actual number, like how old they are. Unfortunately, when I went to remove the bees from the hive, they were not foraging. I think something about being in the tent made them uncomfortable. They weren't really interested. So I was a little bit worried when I was doing my experiment that I wasn't going to see a noticeable difference between the age groups, because they weren't like true forager bees. But you'll see later when I go over my results that there were actually physiological differences despite them not doing these different tasks, or at least not me observing them doing these different tasks. So this was the feeder that I put in the cage with the hope of sort of luring them out and being able to collect them at the feeder, and it didn't work. So these are the cages that I put them in, these little plexiglass cages. They had tape on all the holes, and I also inserted a number of attendant bees. So they were just callow, freshly emerged nurse bees from a different colony to simulate a more natural hive environment, because there's evidence that says that bees on their own, just like 10 bees or something, aren't gonna think that they're in a colony. They're going to have physiological differences. And so I didn't want that to be a confounding factor. So I put some more bees in there just to make sure that they were cozy, basically. So here's a close-up of them in their cages. I had to take care of them every day for about a month during the course of this experiment. So every day I would go in and I would feed them new water. I would give them more pollen and candy. This is them in the incubator, separated based on treatment. And yeah, this is my feeder setup that I was putting them in falcon tubes. There's just little like dishes. So yeah. And then when I sampled them, I would bring them out of the cage and I would put them on dry ice at a variety of time stamps. So the 7th, 10th, 14th, and 18th day. So I would be able to see how some of these physiological differences changed over time. So one of the first things that I did with the lab work was to dissect their hypopharyngeal glands, which are these glands in the heads of the bees. As I said, they produce proteinaceous secretions. Basically, this is where royal jelly is produced. And so you would expect that there'd be bigger and more developed and more full of protein in the nurse bees. And you can see this is an image of a dissected bee. The hypopharyngeal glands are still in the head here. And the hypopharyngeal glands are made up of what are called acini, and this is a physiological term. There are acini like in your pancreas. They're basically just like these little bubbles, and they resemble to me like a nerd's rope, basically, if you've ever had that bit of candy. And in order to quantify the size of the hypopharyngeal gland acini, I used a program called ImageJ, where we would circle the basically 10 of the acini for each bee, and it would automatically calculate the area. And we would find the average area for 10 acini and give that number to that bee. And as you can see, this is a bee that was, I think, a nurse before bee. I collected a few bees before I even did the experiment to sort of get a baseline. And these are really big, they're really developed, and they seem to be quite full of protein. This is a bee a little bit later in the experiment, and they're a little smaller and more shriveled. So you can see there's quite a drastic difference in the physiological development of these glands based on age, treatment, etc. So that was one parameter of hypopharyngeal gland development. I also looked at fat body weight. So I dissected their fat bodies, and I also looked at gene expression of vitellogenin, which is associated with fat body storage and, like I said, reproduction. And winter bees have high levels of vitellogenin hemolymph levels, not necessarily gene expression, but sometimes those are correlated. And I also looked at the protein content in the hypopharyngeal glands themselves, and I was able to compare some of these values. So I'll get into the results now, the exciting part. So what we did was we plotted average acini versus protein mass of the hypopharyngeal gland for all of the age groups. And the reason that I wanted to do this was because, as I said, nurse bees, what really distinguishes them, or what really distinguishes winter bees from nurse bees, is their large acini and low protein mass. So what you would expect would be a nurse bee over here with maybe larger acini, but a low protein mass. And as you can see, there aren't really a ton. There's none, no bees here down in this area. So it didn't look like any bees were here, regardless of control or treatment. But I'll explain a little bit more about this graph. So this is all of the age groups, all of the days, and what we have are the before bees are the black ones. So the nurse bees are the triangles. They had really big acini and really big protein in their, or a large amount of protein in their acini. The young foragers here at the circles before bees, before treatment, had sort of a little bit lower, and the old foragers had even lower in both parameters. And there's a pretty positive correlation between the two. As acini get bigger, or rather maybe as protein gets bigger, acini also enlarge. So that was the general trend, which was interesting. And the sort of spread of data really did vary depending on the age group as well. So we could see that even though the bees weren't actively foraging, and I was worried that they were maybe not behaviorally distinct, there was still a pretty significant difference. So as you can see here, there's the nurse bees by themselves along the same line, and they're much higher, sort of on the higher protein mass side of things. So they're maybe have more active hypopharyngeal glands. The young foragers are a little bit further down here, and the old foragers are even lower. So they have even less protein mass. So this was pretty good evidence that even though I didn't see them actively foraging, the age alone was sufficient to create some pretty intense behavioral or physiological differences, which isn't a particularly surprising bit of information. This has already basically been known, and it's what would be expected. So it was just a nice sort of, I guess, it just confirmed that I was sort of doing things right. regardless of the ethyl oleate. So you don't see really a huge difference between a spread of data points for the ethyl oleate, which is blue, and or control, which is blue, and ethyl oleate, which is yellow. It's all sort of mishmashed together there. So to show this in a different way, we have the before Bs, which are the same in both control and ethyl oleate, because obviously they hadn't been treated yet when I killed them. But the general trend for the acini area as time went on was that they seemed to go down regardless of treatment with ethyl oleate. So ethyl oleate didn't seem to have any effect on the actual size of the acini. And yeah, so they just sort of, there's like a general downtrend. The longer that they're in the cage, the acini sort of start to degrade. Or maybe it's not being in the cage, it may be just that as they age, it starts to degrade, because as you can see here, that the nurse bees started out with higher, and then it gets lower and lower as time goes on. Similarly, you can see the spread of data points is sort of just a different way of showing it, but average acini for nurses versus control versus ethyl oleate was not significantly different. Neither was it for young foragers, for ethyl oleate and control, or old foragers, but you can see that there is a pretty significant spread between the age groups. So nurses were higher, young foragers were sort of in the middle, and old foragers were a little bit lower too. I guess it's dinner time. So the same sort of was true for the hypopharyngeal gland mass. The nurses started out higher and they went down. Young foragers were sort of in the middle, and old foragers were the lowest, and it went down, and it wasn't really affected by ethyl oleate. So as I said, this is sort of the same thing. Nurses were the highest. It looks like it's even a little bit lower here in ethyl oleate, but none of these values were significant, which means that it could have been due to random chance and can't be confidently attributed to the ethyl oleate causing these changes. I need to go over there. So then I did sort of the similar thing. The protein mass to acini area ratio is trying to capture that nurse bee or the winter bee physiology of having large acini but small protein. So you would expect a winter bee to have a lower protein to acini area ratio. So the lower it is, the more winter bee it is. And there was no difference for treatment for young foragers here, 23.1, 23.5. That's not really significant enough to say that ethyl oleate changed anything. 18.4 to 20.3, it actually went up. But in the nurse bees, ethyl oleate did seem to have an effect on the ratio. So it went down, it became more nurse bee-like because of this ethyl oleate treatment, and it was a significant result. And we'll go into a little bit more about what this means in a minute, because I have a little bit more information to sort of corroborate this. Now the fat bodies, they didn't have as much of this downward trend as you saw with the hypopharyngeal glands. It seems to be a little bit more all over the place, but I will show you what happened with the fat body mass. So the nurse fat body mass was significantly increased by ethyl oleate. It went from 0.0037 grams to 0.00425 grams. Young foragers were not really affected, but old foragers were pushed up to the level of young foragers. So their fat bodies got bigger, nurse fat bodies got bigger, and both of those were significant. And then, so wait, where did that go? This was the baseline percent fat body weight for the before bees. So these were not treated. This is just how they came out of the hive. And what this percent of total means is we added up the total hypopharyngeal gland protein mass and fat body mass to get sort of what I call a metabolic output total. So whatever they're eating has to be turned into something. It's getting turned into either protein in the hypopharyngeal glands or fat bodies for storage in their abdomens. And the nurse bees have a lower percentage of fat bodies, so it's still 85% of total, but it's lower than in the old foragers. So the old foragers are spending less time creating, or less metabolic energy putting their whatever they've eaten into hypopharyngeal gland synthesis into their fat bodies instead. So it goes up as they age. Now, how did ethyl oleate affect this? Ethyl oleate basically affected the nurse bees alone. It didn't really affect young foragers or old foragers, as you can see, 96.2 versus 96.5, 96.1 versus 96.4. But in the nurse bees, which we'll focus on, the fat body percentage went from 92.7 to 94.9, and it was significant. And basically what this is saying is that ethyl oleate helped the trend. Normally, that's what you would expect. As they're aging out, they're getting more of their metabolic output going into their fat bodies, but ethyl oleate made it seem more efficient. And I don't think I put this on this slideshow, but nurse bees actually ate significantly less pollen. So they're eating less pollen, but their ethyl oleate is causing whatever little protein that they're consuming, it's becoming more efficiently pushed into their fat bodies than into their hypopharyngeal glands. So I think this is pretty good evidence that ethyl oleate may be contributing to overwintering. It's causing whatever dwindling pollen supplies in the fall to build up more efficiently. It's shifting their metabolism to be more conducive to overwintering rather than rearing brood. So that's interesting. Now, the other interesting thing is I also tracked the survival of the bees, and for a different set of bees, because obviously to sample the bees, I had to kill them. But I left some of the bees alone, and ethyl oleate didn't have an impact on survival. So in this scenario, in the nurse bees, they actually lived less long than the controls. In the young foragers, they lived less long. And in the old foragers, they lived slightly long, like basically the same amount of time. They made it to 60 days, and none of those were significant. So ethyl oleate doesn't directly cause them to be nurse bees, and it's not the elixir of youth, but it may contribute, and it may, with other factors, help overwintering. So I also looked at vitellogenesis. I didn't see any effect from ethyl oleate, but as you can see, there's quite big differences in the different age groups, nurse bees versus young bees versus old bees. They had different gene expression levels, and the same for defensin, which was an immune gene. There was no difference because of ethyl oleate, but between the age groups, there were quite big differences. And this, I looked at this because I wanted to see if part of the mechanism causing like increased longevity was maybe they have better immunity, but now I'm thinking that maybe it's more has to do with metabolism. Ethyl oleate affects the metabolism of consumed protein more than it affects immunity. So the conclusions that I have from this, I basically propose a mechanism for overwintering, which is in the absence of brood, as pollen becomes less available in the dwindling autumn months, they are starting to eat whatever they have, and as it dwindles, they sort of cease brood rearing because pollen is necessary to rear brood. And as pollen starts to dry up, the brood rearing starts to dry up too. And as that happens, the metabolism must shift from hypopharyngeal gland protein synthesis to fat body storage, regardless of ethyl oleate treatment. But ethyl oleate treatment seems to intensify that shift, and it is significant in nurses. So nurses are a little bit more behaviorally plastic. They're able to benefit from the effects more, which I guess makes sense because ethyl oleate is sort of designed to act on nurse bees. So yeah, and then the new proposed mechanism basically is that in the absence of brood, as I said, or brood pheromone, ethyl oleate promotes the physiological arrest of nurses by allowing whatever dwindling pollen supplies that are available to go from hypopharyngeal gland protein synthesis to fat body protein or fat body synthesis. And interestingly, some of that dwindling pollen, as it starts to dwindle, they don't want to feed the brood. They start to actually cannibalize some of their early instar larvae. So some of that protein that's going into their fat bodies is actually not coming from pollen anymore, but possibly from the cannibalization of early instar larvae. So even as pollen is dwindling, they're still getting a good amount of protein to go from instead of into their hypopharyngeal glands into their fat bodies. So what we would need to know for future directions is whether we would want to look more into some of the other physiological markers, such as vitellogen and hemolymph levels. We looked at vitellogen and gene expression, but sometimes those are not always correlated. Sometimes you want to look directly at what's actually pumping through their blood rather than what genes are turned on. We also would want to look at more metabolic genes to sort of understand the mechanism more. I would expect, based on these results, that ethyl oleate can actually change the expression of certain metabolic genes. So that would be an interesting way to look at it more. It's also possible that the sudden shift from my experimental conditions of being in a normal summer hive to being in basically an incubator with a certain amount of pollen every day doesn't accurately simulate the gradual change of the season that prefaces the transition to a diutinous state. So it would be interesting to show like how dwindling pollen supplies affects the interaction of ethyl oleate and brood pheromone and a number of other factors. Because as we know, as biologists, as beekeepers, there's so much going on and so much interacting that we would never really be able to fully like recreate a natural environment when we're managing our bees. So my recommendations based on this is that, well, basically confinement of forage zone can potentially contribute to greater longevity, which other studies from even the 80s have shown that bees in the tropics that are confined to the hive do sort of enter a longer lived state, and possibly due to ethyl oleate. In Northern California, places like this, it may not be good to continually be feeding your bees pollen in certain times of the year, because the brood break is really beneficial to bees, not only to recover from light loads, but also to sort of recover from the heavy energetic cost of raising brood, give them a chance to sort of get their fat bodies stored up a little bit more. And we know that Varroa are feeding on fat bodies. So having a period of time when there's no brood in the hive allows whatever they are eating to go instead into their fat bodies, maybe giving them a little bit more to fight off the Varroa. There's been studies that show that caging the queen is enough to break that brood and maybe potentially impact their metabolism and their ability to fight the bees. Conversely, if there's times in the summer when there's high temperatures and maybe more ethyl oleate coming into the hive and maybe a pollen dearth, if they're not supplemented, the bees may enter a natural quiescence that allows them to sort of ride out the harder times. So that is basically my thesis. And if you have any questions, I'd love to hear them.
Thank you. Thank you, Nissa, for that great presentation. We appreciate it. There is one question in the chat right now. He's wondering if at the end of the blooming season, as winter comes on, if there's more exposure to fermented nectar in the outside environment rather than inside the hive.
Yeah, I don't know about the actual prevalence of fermented nectar. It's just that ethyl oleate is a really common and integral part of the demographic shifts that happen for the division of labor in the hive. And so I think that what we consider fermented nectar is probably way more fermented than is necessary to create ethyl oleate. So they're consuming small amounts of ethanol from directly in the flowers, not even just in like the honey. So it's probably just like you wouldn't even know that it's fermented. It's just enough to create this pheromone.
Thank you. I have a question about surprises. I mean, you began this research
Mm-hmm.
2.5, 3 years ago with some specific hypotheses that you wanted to prove or disprove, and I'm wondering, based on history and what you've learned, what are the surprises and what are the, well, I figured that was going to happen anyways, moments for you?
Right. Yeah. Well, I actually did a second experiment after this to follow up on some of the questions where I looked instead at the age of the bees at the task. So I collected bees that I observed actively sticking their heads in and nursing, and counted those as nurse bees. And I collected bees coming in to forage. And I thought, like this is going to be such an easy experiment. It's so much smaller. I know more than I did before. I put them in the cages and they just like stung each other and killed each other and died. And I was like, I don't know what's going on. And I ended up with this like really small sample size. And I analyzed the data and it was like inconclusive. It didn't really match the data that I've shown here. Even though I thought that I had sort of distilled down the important parts of the experimental design. And I couldn't really have expected that or explain it really. It was just, I guess, how things go. And then in the more like I expected that to happen, some of the negative results that I got, I kind of am like, well, yeah, that makes sense. Like, for example, vitilogenin, I wouldn't expect a huge difference there because, as I said, I already knew that it's more about vitilogenin hemolymph levels than gene expression, and they're not always correlated. So that might have been just like poor experimental design on my part, or it was just there were certain logistical limitations too. Collecting hemolymph is a pain in the butt. So I actually did try that for a while, and I was like, I can't do this. This is too much. So yeah, the logistical issues are a big one.
It's like you needed a whole team, right?
Yeah, I sure did.
Yeah,
And
yeah.
it was like I did this experiment in like the height of COVID, so it was like fully locked down while I was doing this.
Mm-hmm, mm-hmm. And painting 1,000 bees.
Yeah, that
That's
was.
no small task either, right?
Yeah, totally. And it's funny, I've been writing my thesis and getting it all done, and 3 years of work, it doesn't seem like much when it's on like 20 pages. It's not very, I was like, it's only 20 pages. What's going on? So
Which, before I focus on the next two comments, I do want to bring everybody's attention to a reading list. It's an annotated bibliography that Nissa has so generously shared with everyone in the camp that's available on our website. And I invite you to look for it, have a read. There's a lot of really great um, research papers in there. So, what about suburban areas have non-natives that bloom all year round, right, in Northern California? Would this prolong the brood reign?
probably, yeah, they show that if you continue to feed them pollen, they continue to rear brood, which, maybe isn't what you want necessarily. Like I said, having a brood break can be really beneficial to their immunity and their metabolism and varroa. So yeah, I would imagine that having more pollen around is a major factor in this sort of quiescence.
Yeah, yeah. But like you said, caging the queen, right? It's
Yeah, that's
effective.
the way to do it because then they
Yeah.
don't go queenless and you still, get all that.
Hmm. Um, Another comment here. I have some experience with honeybees 40 miles inland from the coast, and during the late summer, during the drought years, there are very few pollen sources, and brood rearing ceases even in the late summer.
hmm
Yeah.
Yeah, that's exactly what I've been saying. And it may be good to let them cease, and it would be interesting. You could paint some of your bees around this time and see how long they live because you may be surprised that they live longer than you would expect, even though it's summertime.
Mm-hmm. The bees brought to the coast resume pollen collecting within 15 minutes of being released. The
hmm
bees left inland managed to survive till the
Yeah.
spring. So,
So you wonder what they're where are they getting the fat bodies though? Is it because they're cannibalizing brood?
yeah, yeah, yeah. Another one. I live in San Diego, and my colonies have 2 to 4 frames of brood through our very mild winters. They have the ironbark eucalyptus trees blooming starting in November through the spring, so pollen and nectar are available. So San Diego doesn't really see a brood break,
hmm
right?
Yeah.
Yeah, yeah. And just some great comments around enjoying your presentation. Very informative lecture. Thank you, and thank you, thank you, thank you.
You're welcome.
Yeah, yeah. I'm going to officially thank you on behalf of the camp, and we wish you all the best in your future endeavors, Nissa. Tell us a little bit about what's in store for you in Vermont.
Okay, so I'm actually currently working already for the job that I'm going to do. So it's part-time and it's remote and it's going to be full-time in person when I graduate. There's this tiny little college in north central Vermont, and it's called Sterling College, and they have, I think, 125 students. So it's really, really, really tiny, and they focus all on sustainable agriculture. They have a whole they have like a draft power animal course to give you sort of an idea, teaching people how to plow their fields with horses and stuff like that. They only have like 4 or 5 majors, I think, like ecology, biology, like sustainable agriculture. So it's all about like changing the paradigms of food systems, addressing all these issues that are facing, climate. They're going like fully carbon neutral. All of the food on campus is grown on the farm there or within a 250-mile radius. And I'm going to be helping with their continuing education program, where they're creating classes on all of these subjects that are going to be available for free to some of their partner organizations all around the world in small farming communities like Bhutan and India and Puerto Rico. And then eventually those classes will be available to the public to purchase. And so I'm helping them develop like an ecology class, an agroecology class, ecological pest management. And then I'm also going to be taking on a adjunct faculty role in teaching their undergrads. I'll be teaching field entomology, introduction to biology, and ecology foundations. So very exciting.
Yes, yeah, that's right up your alley. I'm so pleased for you. Congratulations.
Thank you.
And thank you again for taking time out of your very busy schedule to speak with us at the camp here. Thank you.